Well, thank you, Kendall. It's certainly a pleasure to be here. I have to relate, though, that when I was talking with Kendall about coming here, I said, you know, after 30 years in Florida in the South, I grew up in Indiana, so my blood had thinned out. I said, I couldn't take cold weather in Iowa. And he said, oh, early November, it'll probably be okay. And I was shocked. I was in Des Moines Tuesday night, woke up 2.5 inches of snow, and I had to dig a car out. I hadn't done that in 30 years, and it was a good reminder why I hate snow in winter. So I think we should have fun today because I want to talk about maximum yield. It's always an interesting topic. We can talk about some assumptions, and I'm going to actually try to make my presentation a little short so we can have some feedback and questions. And what I want to cover in this maximum yield is that I think there are 3 key resources from which each you can calculate a maximum yield. And that will be the input of light, the input of nitrogen, and the input of water. And so those are the key things. Well, when I came to some place like Iowa State, I figured, well, to give the seminar some importance, I really should start the seminar with some classical literature. And I pulled something off its shelf at home, Jack and the Beanstalk. I read this to my children when I first gave this talk many years ago. Now it's the grandchildren that hear this. For those of you that may have forgotten the story, Jack and his mother were living in very economic hard times. It's really a current piece of literature, obviously. They were down to just having a cow. So Jack's mother decided Jack should take the cow off the market and sell it. Let me pick up the story from that point. As Jack led the cow to market, he met a man who said, I will give you some magic beans for that cow. The beans looked so pretty that Jack agreed to the trade. Magic beans indeed, cried Jack's mother when he returned. Son, how could you be so foolish? So saying, she flung the beans out of their cottage window. When the Jack awoke the next morning, he saw a tremendous beanstalk growing from the ground and reaching to the clouds. Being an adventurous lad, he began to climb it. At the top, he saw a large castle. Well, for those that forgot the rest of the story, Jack entered the castle, found a big old nasty mean giant, stole his hen that laid the golden egg, and in the process of escaping, murdered the giant. Now, there's many reasons I don't like this story, even though the kids do. One is it portrayed large people in a very negative light. It just didn't seem fair to me. The other thing, it seemed to condone theft as a way out of your economic misery. And as a part of that problem, it was okay to go ahead and murder the person you were thieving from. So it just didn't seem the right message for kids. For agronomists, what really describes this as a fairy tale are those magic beans. Remember, Jack's mother took those beans and threw them out the window, and the next morning that beanstalk went to the clouds. This is the ultimate no-tillage experiment. There was no fertilizer, there was no irrigation, there was no herbicide, no fungicide, no insecticide. And the amazing thing, it was done in the dark. So we should know that that's a fairy tale. But if I told you that they came back 60 days later and saw that beanstalk, there may be some confusion whether that was a fairy tale or not. I hope after today's story, or the seminar, you will still conclude 60 days as a fairy tale. So that's what we're trying to do, is actually lay down what we know, what's required, and try to do reasonable yield assessments. Now, this is indicated, I like the title, Sky is the Limit, when we're talking about maximum yields, so we can get everyone's attention that there's an ultimate amount. But we look, the sky is the one that provides these key resources, and it does set a limit. It's the sunlight. I'm showing lightning. The original source of nitrogen is from the lightning, and precipitation. And these are the 3 areas I want to cover in this presentation. Sunlight, of course, drives our photosynthetic reaction, fixing carbon dioxide into photosynthate. And we can this is an energy-driven process, something people seem to forget. You have a certain amount of energy, and you can only get a certain amount of photosynthate. You can't exceed this relationship just at the biochemical level, but when we put it on the whole crop level, the relationship still exists. And in fact, at the whole crop level, we can use this term of look at biomass increase, which I'm showing on the ordinate, with the cumulative amount of radiation that the crop receives. And commonly, this is done through the season, you get a very linear relationship. This relationship is called radiation use efficiency with units of grams per megajoule. For these peanut lines, it's 1 gram per megajoule. These are very different eras of release. These varieties, these are 4 varieties, very different eras of release and very different growth habits. But within this species and within most species, the radiation use efficiency is pretty constant. And that's an important fact, I think, to remember when we talk about maximum yield. Now, the radiation use efficiency, as shown on the ordinate here, is directly dependent on your photosynthetic capacity. So as photosynthetic capacity of the leaf, individual leaf increases, the radiation use efficiency for that whole canopy increases. It increases in a curvilinear fashion. Now, the photosynthetic rate, these are old units, show you how old this work is. The photosynthetic rate of our C3 species are commonly right here in this zone around 1.4. We go up here in soybean, and you'd say the radiation use efficiency is around 1.2. This is the kind of level of photosynthesis you would measure. For rice, it goes on up, and you're about in the zone of 1.4. Wheat would have the same value. Now, maize has a C4 photosynthesis, so it's out here at 2.2, 2.4, and you go up here, and your radiation use efficiency is 1.7, 1.8. So the variation between species, while it does exist, you can see it isn't hugely different, and the reason is this curvilinear relationship. This says if you increase leaf photosynthesis, you're probably not going to get a huge increase in this radiation use efficiency, biomass capability. On the other hand, if anything happens that causes photosynthesis to decrease, then your radiation use efficiency really drops. So we got to protect these crops from nitrogen loss, water loss, and other factors that come in and will lower the photosynthetic capability of the leaves. What do I do now? We now know really that for most plants, we know what this radiation use efficiency is, and it matches pretty well what I showed you in the previous slide. For C4 crops, maize, sugarcane, and high production sorghum, for instance, it's about 1.8 grams per megajoule. If you tell me how much light you're giving me to a well-grown C4 crop, this is what it's going to be. C3 grasses like wheat and rice, it's 1.4. And for the legumes, based on that graph, it's 1.2. Now we can use that then to get what the maximum yield could be under any light condition. We find out how much light is intercepted through the growing season. That depends on the light coming in and the leaf area of the crop. We multiply it times the radiation use efficiency, sum it over the growing season, and multiply it by the harvest index, the ratio of the grain yield to the total yield, and we'll have maximum yield. It just absolutely says how much you can get. And I'm showing some calculations here for these 3 species. You can see up here at the top for maize. There it goes. The maize, we'll assume that harvest index is 0.5. This is the radiation use efficiency. I'm going to assume something as high as 18 average megajoules per meter squared per day. On the average, that's probably about what you have out here in Iowa. And we have it for 100 days. This is the intercepted radiation, and you have it for 100 days. So this is a pretty long crop that you have light that high. And if you can do that, then the maximum yield is, the units here are 1,620 grams per meter squared. You can move it over 2 decimal, a little over 16 tons per hectare dry weight. If you want to count what the farmers get to sell as water, it becomes over 19 tons gram wet per meter squared. Now, this is a very substantial yield, of course, and it's not too much, actually, though, above what the top yields are now. So this light says you really can't get much more than some of these top yields we see. We can do the same calculation from wheat. Again, 1.5, the radiation use efficiency 1.4. It's grown during the winter where we might have a little bit lower light. I assume 15, again 100 days, and the yields come over a little bit over 10 tons. For your wheat crop, wet, that's 12 tons. Again, maximum yield we can expect based on the light. Soybean, I'm here at 0.5. I'm going to assume 1. The 1.2 I showed you previously happens, but it's a very rare circumstance. Most soybean top out at about 1. Again, 18 megajoules, shorter season crop, I'm assuming 80 days. The maximum yield here is a little over 7 in dry weight, a little over 8 in grams meter squared. So if you want maximum yields based on these assumptions, I think that's what we're looking at, just based on the light. Now I wanted to show you some calculations we did at several locations. This is a number of years ago where we had different locations actually in the world. In tropical Australia, this is in Florida, in Illinois, and in Grand Junction, Colorado. We had their radiation, daily radiation, the temperature, and then we could calculate out the duration of the crop. And this tropical region, as you can imagine, is very warm. This accelerates crop development, so the season is short, and the yields we calculate out were a little bit over 8 tons. This is, in fact, what they get under their best experimental conditions. In Florida, we have a condition where we aren't quite as hot, and we have, is that me? If we have more days of growth, and we get yields up to 10, 11 tons, and as it turns out, that's essentially what was measured in these experiments. Now in Illinois, we were looking at about 11 tons. Again, for these conditions, that's what we got. We probably, if we had more data, we could do this again. This is a pretty short season crop here, so I expect if we did again for, say, conditions in Iowa, we might get a higher yields. The thing that I find interesting is these data from Grand Junction, Colorado. This is a high mountain valley in the western Rockies. It's in a desert, so there are no clouds. So the radiation you can see is extremely high. So the light is really pumping in. It's heavily irrigated, has to be under those conditions, and heavily fertilized. Because of the altitude, the temperature is low. As a result, we calculate out a growing season as 140 to 150 days. Now the input parameters are the same for all these locations. The only difference is the temperature. And you can see what the impact the temperature did in extending out the season. We could calculate in this environment that you would get up to 18 tons. And you can see that that's about what was achieved in this location. These experiments were done in the early 80s. And as far as I know, there's no scientific data published out there that exceed these. These are the maximum yields that really can be documented. Pioneer has done some data, some experiments in their winter nursery, another high mountain valley in Chile. They get the same kind of yields, but it has the same environment. So light is the one that's setting the limit here. Okay, so the light, I think, sets a rigid limit, and we've got to remember that. There's no, as far as I can see, none of these parameters have much flexibility, and we've got to live with it. That's what we're destined for. Let me then move on to that second parameter, the nitrogen limit. Now the lightning would give something maybe 10 to 40 kilos of nitrogen per hectare per year. This depends on where you're living. In Florida, where there was a lot of lightning, it was on the high side. And of course, if you're in a desert where there's no lightning, it will drop to 0. This is the input on which the native grassland species in the Midwest grew. They were able to sequester it and maintain it in the soil. But this was the key nitrogen input. And you will see that most of the yields through history are tuned to this kind of input. Okay, so this is what the input was through most history. But obviously now we put on fertilizer. And why do we need all this additional nitrogen? The first thing, every new cell requires nitrogen. You can't have nucleic acids, you can't have protein without nitrogen to put in there. So you have to remember, if you don't give that plant that nitrogen, it can't form a new cell, and so it can't grow. So it's absolutely essential to have that. The second thing is the metabolic activity is dependent on those proteins that have the nitrogen. If you don't have those proteins operating, you obviously aren't going to have the same metabolic rate. And I'm going to show you the impact on photosynthesis from this. The key, though, is you also need the nitrogen to support the newest tissue. If you're not taking up the nitrogen, nitrogen's pretty mobile in the plant, it's got to go to those new tissues. And if you remember, when you have grain production, Usually you have very little nitrogen taken up, and this is the key. You've got to move this nitrogen from the old tissue into these new tissues in the seed. So that's a key part of that. So you need a lot of nitrogen to match the nitrogen that you're putting into the seeds. Well, I wanted to show you a bit about the photosynthetic activity in terms of nitrogen. On the ordinate here is the assimilation rate, and on the abscissa is the leaf nitrogen content down here in grams per meter square. Here is maize, and here is sorghum. And what you'll see is that this is the number of plants data taken over a range of conditions. We had to get this variability in leaf nitrogen content, but all the data fall on one line. And if you go to literature and look at other data from maize, this is what describes it. They are very conservative within the species. So this is a kind of factor for nitrogen. Sorghum, of course, is another C4 species, and it has its own curve, not too much different from maize. So that if you tell me, in fact, I've gotten to the point, tell me how much leaf nitrogen is in that, or nitrogen's in the leaf, and that'll tell me about photosynthetic capacity. Don't waste my time with this going out in the field with these chambers and try to measure it. It's just too hard and too variable. This is the one where this shows you what kind of variability you can get. Okay, well, I want to go back again to radiation use efficiency. This describes the capability to accumulate mass. And what we've got plotted here on the ordinate is the radiation use efficiency. And because we know it's a function of leaf photosynthesis, and leaf photosynthesis is a function of leaf nitrogen, then we can make this direct plot. And what you see, this maize is very responsive to increase in leaf nitrogen. The radiation use efficiency increases dramatically. Probably no surprise why maize was the target for initial studies on nitrogen fertilization. The other thing that's more obvious here than it is in photosynthesis is that it saturates here. That you cannot push this, even with more leaf nitrogen, to higher radiation use efficiencies, and this tells you why we're in that zone of 1.8 or maybe even 1.9. But this is the limit. If you pretty well stoke up that leaf for maximum photosynthetic activity, this is what you would get. Now rice, the C3 species, has a different curve. It's offset a little bit, and its increase is slower. It goes up here, and again it comes to about this 1.4 zone for the amount of leaf nitrogen you can put in the rice plant. And soybean is again, it's offset from rice. There's a lot of storage in nitrogen in these legume leaves, and this apparently is the basis for this offset. It seems to me there should be a way we could move soybean lower so it isn't so sensitive to all this nitrogen, but in fact most of the photosynthetic data follow on this curve, going up to as high as 1.2. So the amount of nitrogen that we can pump into the leaves tells you about the radiation use efficiency, and that tells you about how much crop we can produce. Well, what can we do to overcome the nitrogen limitation? We always hear, it seems to be extremely popular now, say, well, we're going to get the more, they use the phrase nitrogen use efficiency, which drives me nuts. I don't know what the hell that means, but they like to use it. As far as I can tell, when I look at the physiology, one thing would be to improve the uptake pathways, get more nitrogen out of the soil at the different concentrations. You have to remember, these plants evolved in millions of years always under low nitrogen. And there's multiple pathways, some with very low affinities for the nitrogen in the soil. There's no, as far as I can tell, there's almost no evidence that this is a limitation in uptake nitrogen from the soil. The pathways are there, and they're ready to be turned on if the plant can store that nitrogen. We also hear about efficient use of nitrogen in biochemistry. Again, the plants have evolved in millions of years, and they are very conservative with this nitrogen. I just don't understand where the biochemical process is that's wasteful of nitrogen. They're very conservative and frugal with the nitrogen, and I don't see a target zone for this. The challenge in our crop production is not at these levels, but to get a lot of nitrogen stored in the tissue during the vegetative growth so it can be available when the seeds start growing to support that seed growth. And that's the key in trying to increase the yield in terms of the amount of nitrogen you have. How can I get more nitrogen in that plant so it's available when we're growing the seeds to support those seeds? I wanted to show you some calculations I've done recently where this is the amount of nitrogen essentially can be stored in the plant in kilograms per hectare, and it's based on the crop mass. As the crop gets bigger, obviously it has more tissue to store away nitrogen. And these relationships were actually worked out based on nitrogen concentration in the stems and leaves. So I've taken these numbers, rearranged them, and we get these responses. You can see for maize, if we wanted to store over at least 200 kilos per hectare in that plant so we can have it for the seed, we've got to get a mass before the seeds start growing of about 16 tons. You have really got to push in a lot of mass to get that nitrogen stored. The wheat is a little bit better off. It has more nitrogen in the leaves in particular, and you can see that you still need something like 10 to 12 tons just to store this nitrogen. The legumes, remember I was talking about soybean having all this protein in the leaf. The advantage is you don't have to get so much mass to store that nitrogen. So one of the real spreads in our species is how much nitrogen they can store, which they'll use to grow those seeds. And to me, this is the really challenge, is how can we maximize nitrogen storage in our crops during their reproductive development. Now I want to take a little aside and talk about one trick that we've employed, but I don't think most people recognize it. And the trick was that we used went to erect leaves. And the reason this is important is because the leaves at the bottom of a canopy aren't parasitic. And that if they don't get a little bit of light to keep them going, they senesce and fall off, and of course they take all the nitrogen with them. So you need to retain all these leaves to get a high leaf area index just as nitrogen storage reservoir. So you've got to get a little bit of light down there to keep them going. And the way you do that is to get erect leaves. So we can do the calculation. This is for rice. Up here on the top axis is the yield of rice. And every increment of yield, of course, is directly related to the amount of nitrogen you need, the percent nitrogen in the grain. So this translates directly to the nitrogen needed for the seed. And that amount of nitrogen stored, it translates into a leaf area index. Okay, so how much leaf area do you need during vegetative growth to store the nitrogen to get these yields? And what we see is to achieve these leaf area indices, we have to have more erect leaves. So our old crop, where we had the lower yields for rice, is around 4 or 5. A flat horizontal leaf of 30 degrees was fine. You could allow larger enough leaf area to store that nitrogen. But when we try to push that the yield higher and get more leaf area to store that nitrogen, then you're moving up to leaf angles, say, of 60 degrees or so. And I think this is one of the things we see many of our modern crops with these erect leaves. And I think the reason is that this allows you to pack in a lot of leaves in a per unit area to store nitrogen so you can support the seed growth. Now there's been going around that the reason for erect leaves is photosynthesis. The data is virtually nil or very small that photosynthesis can benefit from these erect leaves. The benefit of erect leaves, I think, is in the nitrogen storage. Oh, shoot. So let's calculate the yield. We can calculate the yield again by the amount of nitrogen we have in the plant, and we put it in the grain. And that gives us a direct estimate of the maximum yield. Here's the amount of nitrogen that's stored in the plant, ready to support the growth. This is the nitrogen harvest index. This is the fraction of the nitrogen you accumulated that you can send to the seed. This is very high now, usually about 80% of the nitrogen. And remember, the vegetative tissue has structural proteins, so it's not likely we can push this much higher. This is the fraction of nitrogen in the grain, the percent protein or percent nitrogen in the grain, if you will. So if we know that this amount of nitrogen, this percent in the grain, the nitrogen accumulated, we can calculate out the maximum yield of any crop. And here what we have is the nitrogen fraction. Rice is about 1.3% or 0.013. In these calculations, these were done a while ago, and I used 0.016. Now this number's dropped dramatically, and I think you can see that if that's lower, that's going to give you increased yield, and I'll talk about that a bit. And wheat then is a higher protein seed, and it's up at 0.022. So I have the fraction of grain, the amount of nitrogen stored, the nitrogen harvest index, and then this is a plot then on the ordinate is the maximum grain yield we can expect, tons per hectare, over the amount of nitrogen that we've taken up and stored for the use by the crop. Remember, we were talking about can the maize crop get to about 200 to 250. If we go up here, we're looking at yields, this is dry again, approaching 12 tons. We're approaching the maximum yield then, just on this ability to get the nitrogen. If we go on up, the rice has a little bit less, and we're maybe talking about 13 or 14 tons. Now the soybean, if we were in this zone, it'd be in terrible shape. It'd be 2 to 3 tons. Fortunately, soybean can store a lot more than that. It has these high protein leaves. Remember, I was complaining about the protein in their photosynthetic rate. Well, it's fortunate they have it because that's what they use to get the higher yields. So those high nitrogen content of leaves are crucial in getting the high crop yields. So one thing I would point out then, this is a linear relationship. If your nitrogen fertility drops down, you're going to lose yield. We don't have it. There's no debate about that. You've got to fertilize or have the fertility to get to these different yield levels. And the problem is to get it any higher, it's not applying fertilizer, it's getting the plants to be able to store it. And that's a real challenge. And I'm not sure we can do much about that. Oh, shoot. Let me show you how this all relates to the recent yields in maize in the United States. On the ordinate here is the yield. This is the national average. And here on this on the abscissa is the year, starting in 1950. And you can see starting at 50 through the early 70s, there was a large linear increase. I should say most people just draw a straight line through all these data. And I'm not sure how you do that, because it looks to me there's clearly a break. And we have one slope from 1950 up to the early 70s. The yield increase per year was linear and quite dramatic. But after that period, the yield increase dropped almost half. So in recent years, we're at much lower yield levels. And what we'll see here is that this earlier ramp up was associated with increasing fertilization. The farmers were putting the fertilizer on, the breeders gave us bigger plants that could use it and store it, and we could get these yield levels. Since then, the amount of nitrogen put on, that's shown here on the right ordinate, has pretty well stabilized. Now, why do yield keep increasing? Well, I think there's maybe two reasons, and both of them related to nitrogen. One, the seed grain nitrogen's dropped. So we're kind of cheated. We're getting the higher fertility just since we let the grain nitrogen drop. So as you saw in the previous equation, that translates into a yield. The second one, I expect we're doing better nitrogen management. Certainly the farmland is being concentrated into the hands of the richer farmers, usually the better managers, and we're probably getting better nitrogen management. Now the question is, how much further can this go up? I mean, DuPont and Monsanto say we're going to go to 300 bushels, and here we're down to about half that. They're going to double that, and the problem is, remember I was talking about how much nitrogen you can store, and that won't take us up much further. And if we put water on top of this, I'm going to suggest that the maximum yield for this country is probably going to top out something like 10.5 to 11 tons. Now, I'm safe in saying that because that is about the life of my life expectancy. So I expect to be dead before I'm challenged. So some of you younger people, you can remember this and claim that Sinclair didn't know what the hell he's talking about. But it looks to me that we may be in due for another break in the next 10 years or so. Okay, that gets me through nitrogen. So we've shown how light can set a limit. We brought the limit down a little bit with the fertility because of that challenge of storing it and using it. In our current cropping situation, it's water. Water is what really limits our yields consistently. Now, what I want to remind you is why water is important is when conditions are right for photosynthesis, the stomata open so the carbon dioxide can move inside the leaf. But when the stomata open, the water vapor moves out. So here we have two gases moving. The physics are the same. They're moving through the same portal. So for every molecule moving in, we know exactly how much water moves out. So there's a very close, intimate relationship about growth and water used. And this has been recognized, well, actually it's been recognized for 100 years. There was a large number of experiments in U.S. and Canada and Europe around the early 1900s where they grew crops and looking at the water used and the growth. And DeWitt, in a classical analysis in 1958, took all these data and separated them by species. And for each species, he plotted their growth here on the ordinate against the water used normalized for atmospheric conditions. And that normalization was all these pots. Experiments had open pots with water in it, so that gave him an estimate of the atmosphere. So he used that to normalize the data. And what he did is just plot it for each species. He didn't worry about what was the variety, what was the fertility conditions, what was the watering regime, what was the cultivar, any of these things. He just plotted it for the species. And what you can see is there's a very close relationship between this growth and the water used. A linear, very tight linear relationship for each of these species. Goes back to the physics of water. You cannot gain growth in CO2 without opening it and losing water. And this actually happens to be sorghum. B was wheat, and C is alfalfa. We actually now know this relationship in a mechanistic sense over what DeWitt offered us. And the slope is what we're calling this coefficient of transpiration, a KD. So the relationship is really that growth divided by transpiration, KD. And in this case, we're using the atmospheric vapor pressure deficit. This actually describes the water gradient that the water vapor is moving through. So this is the relationship. We know what KD is. It's actually dependent on a lot of terms. But all these out here are essentially constant. Well, this is atmospheric CO2 concentration. So this is going up. With this going up, KD is going to go up, and that slope's going to go up slightly for us, which means good for us. We're going to get more yield for the water we have. But the two variables that really count are this B and C. B is the photosynthate conversion to plant mass. If you're producing a crop with a lot of starch, for instance, then you're getting a lot of mass for that photosynthate. So the value of B is high. Okay? And if you're producing a lot of energy-dense material, lipid and protein, for instance, B is low. So this B determines that product that you're producing. C reflects really it's the internal CO2 concentration, that gradient of CO2. This was for the gradient of water vapor. This is for the gradient of CO2. And interestingly, the stomata regulate to keep the ratio of the CO2 inside the leaf as a fairly reasonable fraction of the external CO2. And for C3 species, this is about as high as goes to 0.3 or a little more. And for C4 species, it'll be 0.7. So that you can see the C4 species have the advantage of making B higher. So if you had a C4 species, this would give you a high C. If that species was producing a lot of starch, the B value would be high. B and C high, the KD would be high, and that's what sorghum was. It had the highest slope, and this is the reason. Now, if you're producing that starch, but you're doing it by a C3 species, then this term is lower. So the KD value is lower, the slope's lower, and that's what wheat was in the previous slide. And if you have a C3 species, but you're producing a lot of energy-dense material, a lot of protein, for instance, then both B and C are small, KD is small, and that's the low slope of alfalfa in the previous slide. These equations actually predict out what the water use efficiency is, or this water use for each species. If you know these terms, you can calculate out what you expect. This has been studied in several experiments and confirmed that essentially these are pretty well in the right neighborhood. And we know now that that KD is for maize, sugarcane, the C4 species, is about 9 pascals. It's kind of a hard number to get your head around. It's in pressure units, but that's the way all those things combine to come together. So we get a high value KD, a 9 pascal. For the C3 grasses, it's about 6 pascals, and for the C3 legumes, it's about 5 pascals. This tells us how much water we've got to have to grow these crops. And again, I don't see any way that we can change this. This is the facts of life in terms of getting water by the crops. We can use this to get maximum yield again. Based on water, we multiply the harvest index. Well, let me start over here. If we know the amount of available water for transpiration, then we multiply it, by the KD divided by the vapor pressure deficit, and multiply it to harvest index, assuming that's 0.5. And in this calculation, I'm going to show you, or assume, that the vapor pressure deficit is 2 kilopascal. That's probably about the range for Iowa during the heart of summer. So here we can calculate the maximum value based on different amounts of water for transpiration. And that's what this graph is. The ordinate is the maximum grain yield, and the abscissa is transpiration. Now, transpiration is not total rainfall, because you're going to lose some water through deep percolation, runoff, and especially soil evaporation. So you got to put that aside. The water left for transpiration is what determines yield. And you can see here that, say in the Midwest, we might have a good year, something between 400 and 500 millimeters of water. We go up to maize, and you see, okay, we can get about 14 tons. So it's actually a little bit more than we would get based on the nitrogen calculation. But they're about the same, that we're now fertilizing to match this high water input. The rice is down here at about 8 tons. That's a good yield for upland rice. The high yields are always under paddy rice, and they're blowing this curve way out here because they have unlimited water. So that's what you'll see. And people talk about making upland rice to yield like paddy rice. Well, you can't do it unless you irrigate it. I mean, it's just a foolish argument in some sense. Soybean is in pretty good shape because it's here in the yield range of 6 tons. So these are the kinds of yields you'd get under good conditions. But we don't get this every year. And not many years it'll come down, and you're sliding down. It's a linear relationship. And so the maximum yield for that season is much less than what we would calculate in the high rainfall year. And people talk about increasing yields in zones, for instance, in Africa, where the rainfall is down here around 100 to 200, and they talk about this doubling the years, getting these high yields. You can't do it. There's no water there. You've got to have water to go out the stomata to get them to open to get the CO2. Well, we wanted to know in the United States what would be the yield on a year-to-year difference across the United States. We did this with Pioneer Hi-Bred doing these simulations. I used our simple maize growth model to do these calculations. The growth is like we showed earlier in this talk. It's based on the amount of light intercepted and the radiation use efficiency. We calculate the transpiration. And the transpiration, of course, is a response to growth. However much growth you have, then you know how much transpiration. I should say, by the way, for any of you that are interested in modeling, most of the models have it wrong. They calculate transpiration by meteorological functions and ignore the fact that the transpiration and growth have to be very intimately linked. And if your model doesn't have that, then you have a problem in really showing what the water use is. We have the growth and the development of the crop dependent on soil water. I'm calling it fraction of transpired soil water. That's volumetric water content. And these have unique functions, and we can calculate out the growth dependent on the soil water. Pioneer Hi-Bred has this amazing database. They've divided the U.S. into 30 kilometer by 30 kilometer grids. There's 265 grids in the U.S. for soybean. And for each grid location, they have 50 years of weather data. They've constructed the weather data either based on observations in that grid or through kriging, I think is the term where you use ways to interpolate into 2 dimensions. They have a layer with soil and a layer with the sowing date. So we would run the model, did run the model, for every grid location and every year. So that was over 130,000 runs of the model just to get the background data. So this is the old Pioneer computer was groaning a few nights when we asked it to do all these calculations. This is the kind of results we get. And what I'm first going to show you is I'm not going to show you mean yield, because I don't think mean yield is very helpful to us anymore. What I want to do is show the distribution of yield, and these are the percentiles of, in terms, at each location we would rank the yields and then divide it by percentile. And this top one is at the dry end, and this is the 25 percentile. This is the median year, and this is the 75 percentile, the wet year. Now you can see towards the wet side, well, the blue, the color code here defines the yields in grams per dry matter. And you can see you can pretty well pick out this corn belt. It is the area with the darker blue. On the wet years, it gets these higher yields. And we're in here in the zone of about 1,400 grams per meter squared grams per meter squared dry, 14 tons. That should sound familiar from what I just showed you. But if you go just to the median year, now this wouldn't be recognized as a dry year in the sense that the crops were wilting or anything like that. This is a median year. You get it in 1 year at 5, and you can see the yields dropped everywhere. We don't have this deep blue. The corn belt is still pretty well defined, but the yields here are dropping down to 10 and 11 tons. Now this is a dry year, and I think most people would recognize it as a dry year. The farmers would recognize it as a dry year, but you can see the yields have dropped dramatically. And these are maximum yields in, so the maximum yields in these dry years, we're talking about in some of these locations being very low, where we're talking about 8, 9 tons. So the maximum yield is how much water you get. And I think much of the year-to-year variability that we now see out there in the field is defined by how much water you have. Okay, well, I've gone through each of these. What I hope to show you is that light sets a maximum yield. It's just the energy to drive the process. Nitrogen must be available to grow those seeds. You can't have seeds without the nitrogen. And ultimately, it's the precipitation that allows you to gain the carbon dioxide that is limiting yield and defines the maximum yields for the crop. That's the end of my prejudiced thoughts, and I hope we have some time maybe for some feedback, questions, or comments about what maximum yield can be. Thank you very much. There's no questions? Oh, shoot. The heat. Well, there's a lot of confusion about the heat. First of all, physiology of our plants, corn plants included, is pretty tolerant of heat. It takes really high temperatures to probably have much impact in normal physiological process. Now, I don't know about pollen growth and that. That may be another thing. But the impact of high temperatures is the development of your crop is very dependent on temperature. Okay, remember that slide I showed you, what happens under high temperatures? The season is very short in the tropics. And that's because it's high temperature and it speeds that crop through its whole life cycle. So high yields are going to give, or high temperature is going to give you lower yields. But it's mainly because of the development speed that you put on these crops. And I'm not sure that temperature. The other thing, a high temperature is that it increases the vapor pressure depths of the atmosphere. And you saw how difficult what vapor pressure did. So you're going to use the water fast, and it probably will put you in water stress. So high temperature is not good, as most people recognize. But unfortunately, many times they say it's for the wrong reason. Okay? I've noticed that.
As far as I can tell, what you said is that they've reduced protein content of seed.
In maize.
Maize breeders, I'm talking
Oh, okay.
Maize breeders work hard.
The rest don't, maybe.
They've reduced the protein content of the seed, and they've made leaves more upright so that they can retain leaves as a nitrogen source for translocation of seed. Yeah. What else have they done?
Well, they made a stronger stalks, generally by making them shorter so they can handle all that nitrogen and the grain weight. I don't know.
So if they just directly selected for lower nitrogen seed content and more leaves with retention and strong stalks, would they not work?
My hypothesis, they'd probably been the same point. And I've irritated some breeding audiences because that's a pretty narrow view of what breeders have done. And what we've seen, the major contribution, other than those things, has to do with the disease and insect problems. That is really what we're fighting out there in many cases. You know, I conveniently ignored all those problems. But a lot of the breeding is defensive breeding. And as we move to these higher levels, you're serving a banquet to these pests. And the challenges are even greater in this defensive breeding. So it's hard working, and the job in some ways is going to be harder. Yes?
What about different changes in root to shoot ratio? It's really low in corn compared to other C4 grasses.
Well, for annuals, it's not. I don't think it's that different. But I'm not one that I don't challenge it because I'm not going out and dig roots. I've done it enough, and I'm finished. Usually the root fraction on most of our crops is less than 20% of the mass, commonly less than 15%. I don't know that really enters into the whole question of increasing yield because it's such a small amount. The problem is those roots don't store any nitrogen either. So they're a loss in terms of nitrogen storage. Yes. No, it's related to the fact that they both of those are dependent on photosynthetic pathway, C4 pathway that lets them produce more mass with less water and more on the less light, and the product they're producing starch. So that's why both are higher. Now, as a result, because both are higher, the water needs of corn are not any less. I mean, the calculation is it's more efficient, but it's producing all this mass. So the water needs are equal to or more than soybean, for instance. Did I answer your question? Or?
Not exactly. I guess I was wondering if it related to possible differences in pore density in leaf material among species. I don't know, obviously they're different enough where...
Well, I don't... Well, we could look at the leaf. Yeah, the leaf area is an important term, mainly for storing the nitrogen. So certainly look at that. But I'm not sure. I mean, we pushed all the crops to high leaf area now, so we can talk about more. I mean, it's an intriguing idea. Somebody, go here.
Some, as you pointed out, some people are arguing that they can generate yield much higher than the numbers you propose. Unless they're just making the numbers up, they must have alternate models.
No, they're making them up. Monsanto is making the number up, and they make it up in this great faith. It's a religion, the religion of molecular genetics. Because I've challenged Rob Fraley directly in front of an audience, and his comeback is, "Oh well, we'll stack all these genes. We got all these genes. We know when we put all these genes together in the right format, we're going to get these high yields." It's crazy talk. They're giving you crazy talk. He's interested in selling stock. And unfortunately, his effort to sell stock is killing research priorities, in my view, field agronomist. Okay? Yeah, don't just discount these companies. They're selling stock and they're not talking to you.
So one thing that has happened with corn hybrids is that modern hybrids,
Well, with greater plant density, you're getting the leaf area up. But you can't do it unless you don't shade those bottom leaves. So the density's gotten up by putting more plants in there. And you have to do it by probably having these shorter plants. You don't want these big things that'll lodge on you. And you got to have the erect leaves so you get the light down to hold that leaf area index of 6 until grain fill. So the erect leaf, the planting density is important. It has to go along with erect leaves. At least that's my perspective. Do you have an alternate view? Okay. I think he does. You ask him later.
Well, I'm just a plant breeder. All I know is it's either got more erect plant density going on.
Well, of course, that's what I'm arguing. That's exactly the output you want. Yeah.
Yeah, our pea industry, field peas, has gone primarily from foliage peas to semi-leaf, which is a lot of fine leaves.
Yeah.
Like they're tendrils.
Yeah.
And after collecting data on peas for 15, 18 years, I wish I had some higher reception peak content. And the end concentration tendril.
Yeah. Well, I've worked with some folks in France on their field pea work, and in fact, we've applied this model to that. There's nothing special about peas. It fits in all the arguments I've just shown you. Well, it has a little bit different radiation use efficiency. It doesn't put as much nitrogen in the leaves as, say, soybean does. So those things, once you take account for that, then everything works out the way I described. Yeah.
Max, what about nitrogen inputs from the lighting? You just listed that input and not microbial inputs from fixation. Do you know how they compare?
I don't have an answer for you. I mean, it's a good point, and I don't know there's good data out there. If there is, I'd like to know about it. It depends on who measures it and when they measure it. Oh, sorry. The question is how much is microbial fixation of nitrogen in the soil contributing to the soil balance, nitrogen balance? I just don't know, and I think it's quite variable. Depends on the soil water conditions and everything else. But I think the total amount is, it's less than generally what lightning is. Of course, was in Florida where lightning is really pumping it in. Yes.
Your data present hybrids that are of a non-traded era.
I'm sorry, there's noise here. I missed your. Could you repeat it, please? There's noise.
So the hybrids and the data that you presented are from a non-traded era, but today's hybrids are traded to the max. Farmers have taken them and run them. And so I guess my question is, how do you account for the yield gains seen with the traits? The rootworm protective trait will probably help the roots absorb water and nitrogen. Molecular markers work for disease and probably going to improve radiation efficiency through the air. The water transpiration will be improved with water tunnel.
Yeah.
So how do you
Remember my comments about don't believe these seed companies? I want to remind you of that. Because most of these traits I don't think you can document contribute to yield increase. The yield increase I've seen since they've introduced most of these molecular things that you've ticked off, the main one has been you put less nitrogen in the grain. They're playing the trick. They're letting you get more mass with the same amount of nitrogen.
So on that line of thinking, then conventional corn would be doing as good as traded corn.
I think that in most cases that would be. It's harder to do because you have to make sure you control these insects and control the pests. The comfort, these molecular products are very attractive because they make things comfortable. You can get these things with just buying this transgenic and doing what they recommend. But there's evidence that some of these are lower yieldings, that they're not higher yieldings at all. So maybe if no message other than be very cautious of what these companies are claiming. They're selling stuff. They're not. It's not science. Oh, no, golly. Let me go on this side then.
So why couldn't he come back to you and say, well, we're gonna put these transgenes in, and what we're gonna do is we're gonna speed up the growth rate so leaves are initiated a little bit faster, and we're gonna end up with more leaves and grain filling, and we're not gonna lose those leaves. We're gonna use transgenic approaches to achieve those goals. Would you?
Well, I mean, it would help if you can somehow magically retain leaves that aren't getting light. The leaves aren't parasitic, and they've got to have that burst of light every day to keep them going. It's just to keep them in a subsistence mode.
That's reasonable because there's a signal that's going from a leaf, and the plant is letting it senesce, and we can interrupt those signals.
I think it's more involved in that. You've got to sustain the chloroplast in that, and you've got, and somehow, if you're going to sustain those, you've got to have material coming in. So you've got to overcome this whole idea that they're not parasitic. So all you're doing, but anyway, what you're doing is giving the ability to store more nitrogen. It doesn't get away from their killer on all their arguments is water, and the only solution to that is irrigation. So unless they're going to irrigate the whole U.S., maybe that's their plan, their devious plan. They're going to sell irrigation equipment.
Well, they said they were also going to keep inputs per unit grain constant. So.
It's. Remember, where's my story? Remember that guy that sold magic beans? This is what I was talking about.
Any other questions, comments? Okay, let's thank Dr. Sinclair.
A full list of agronomist podcasts are available at the Department of Agronomy website, www.agron.iastate.edu.